Hello, everybody. My name is Liam Good. I work at the Royal Veterinary College, and I will speak to you today about our project, non-antibiotic control of ETEC in piglets using phage and polymer strategies. This is part of the H2020 funded project Avant, and it's work package 2. Here's the work package overview, and we're looking at developing phage and polymers as two separate strategies, but both non-antibiotic strategies to control ETEC in piglets. And as many of you will be well aware, ETEC in piglets is a very large area of animal health need where non-antibiotic strategies would be beneficial. So drilling down into the more specific objectives, we aim for both strategies to determine the potency and spectrum of activity. That's very much in vitro work, develop the safety information package, assess the clinical efficacy and safety in piglets, and then assess the target specificity in vivo with microbiome analysis following the in vivo efficacy studies. This is quite ambitious objectives, especially given that they're two different strategies, but we've got a good team working well together. People at the University of Copenhagen, the Royal Veterinary College in London, SFR in the Netherlands, ETEC Care in Denmark, and Vet in Germany. So let's look first at the phage approach. Most of you will be aware that phage are effectively bacterial viruses, and they can work quite aggressively, really, by destroying the bacteria by specific species recognition and then lysis of the bacteria. What the team in Copenhagen is then doing is looking at the pathogens that cause the diarrhea in piglets, the ETEC strains, and isolating phage and learning how to propagate them and grow them up, effectively manufacture them in a way that they can be used in an animal study. So there's a lot of early stage microbiology involved and a lot of development involved in scaling up the production of the phage and getting high titers of the phage, which you can score in vitro, but they need to be high titers in order to have an effect in the animals. So far, the results in vitro are indicating a growth effect against the target bacteria. It's modest at the moment in liquid, but the team is working to amplify the phage, get higher titer, and improve the growth effect. Parallel to that, in order for this approach to work, you really need to be able to quantify the number of phages and quantify them not just in sort of clean microbiology media, but also in feces and perhaps in gut content. And so to do that, they're working out methods, which are molecular methods, as you would probably guess, which can be applied in those different matrices. And if you're interested in the details of that, it's been published here. Soon, the team hopes to be putting the phage into animals and testing the effect on diarrhea. So now let's look at the polymer approach, which is the other strategy against the same disease. And again, it's a fairly natural strategy in the way that the polymer we're using is obviously a purified chemical, but chitosan is derived from chitin. Chitin is the second most abundant polymer in nature after cellulose, and chitosan is widely present in fungi and shellfish and the cell walls of many different cells. So we're using a fairly natural polymer here. And then if you look through the literature, you find many reports where chitosan has been used to look at control of diarrhea and promotion of pig health. And so here's a forest plot, and you can see that most of the studies are indicating an advantage. We've looked quite deeply at these, looking at the effect on growth and diarrhea. And from that, we've decided to look at further at a particular chitosan called chitosan oligosaccharide, which is a short version, small molecular weight. And that's based on the sort of positive results from the meta-analysis, also the easier chemical analysis of the shorter molecule, and then that we can access a GMP feed grade. And as you would imagine, we are busy with the microbiology and the chemical analysis, and that will all happen ahead of animal studies, which we hope to initiate this summer. Here's a quick view at the setup of the animal studies, and the two key points I wanted to make was to give you an idea of the percent in the feed, which is quite low, but the forest plot indicates that will be sufficient for an effect. And then these are the zootechnical parameters with a very close look at the efficiency, and then also the spontaneous diarrhea level. We did run into somewhat of a surprise early stage in this project, where we had anticipated to see quite strong or potent antimicrobial effects against the ETEC strains when looking at the cost based on the meta-analysis. But in fact, we didn't. But nevertheless, the benefit seems to be there. So we've developed two alternative hypotheses, which is less antimicrobial as mechanisms, and rather we're thinking that the chitosan is flocculating the cells, or perhaps interfering with the bacterial adhesion to the intestinal cell walls. And these are being tested as hypotheses. Either of them would explain the benefit that's observed in vivo. So to support the studies, both the phage and the polymer, the animal models are being developed, both the growth assays and the challenge models. The growth development models are being run in the Netherlands, and the ETEC infection model is being developed and run in Copenhagen. Lots of acknowledgements here. It's a challenging project in many ways, very exciting. Just to remind you, the teams that are involved in this work package are in the Netherlands, SFR, University of Copenhagen, Klifavet in Germany, Easy Agrocare in Denmark, and then the Royal Veterinary College in London, where I work. Thank you very much for your attention. I look forward to questions and follow-on discussions. Thank you.